Okay, it is 2 o'clock, and so we'll get started on session 3. Hello and welcome to session 3, Trends in Campylobacter Epidemiology and Public Health. My name is Nicole Ginecco from the Quadram Institute Bioscience, and myself and Andrew Grant from the University of Cambridge will be chairing this session with the help of Lisa Crossman and Ricardo Castillo. Now I will stop my share, and Fran, we will introduce you and you can share your slides at any point now. So our first speaker is Fran Colles from the University of Oxford.
So thank you very much for doing such an excellent job of organising the conference and for inviting me to speak. And I think I was rather ambitious trying to cram 20 years worth of research into such a short talk, but I'll give it a shot. So when we started the research 20 years ago, Campylobacter had a reputation for being genetically unstable due to the serotyping methods that were available at the time. So we developed in 2001 a 7-locus MLST typing scheme to index genetic variation across fragments of the 7 housekeeping genes there. And we also at the same time developed the PubMLST database. So this contains all of the sequence typing information, STs, clonal complexes, and so on in the typing part of the database. And we also collect all of the metadata. The database is curated, and we relaunched it last year. So there's so much functionality on the database. We have developed a YouTube channel, and we're collating talks on PubMLST, and also developing tutorials to use the database to its best advantage. So nowadays we of course have the whole genome sequencing era. So we have the core genome MLST schemes, so that, as Nicole said, there's some discussion going on there. Of course, it's entirely compatible. We can still generate the 7-locus STs in silico from whole genome sequencing. And that's still relevant today. We're still using the nomenclature. And yes, it's not restricted entirely to strain typing. You can make bespoke schemes if you're interested in AMR, loci, and so on. So very early on, we realized that with the sequence type data, we saw clusters of very closely related sequence types, which we called clonal complexes. And they're named after the central genotypes. So this was an early publication showing on the right, for example, 2 clonal complexes, 177 and 179, which are associated with wild birds, but in this case are from contaminated beach sand. 257 towards the left there would be an example of a chicken-associated clonal complex. And then also there's these 2 clonal complexes, 21 and 45, which have wide host distribution and a more generalist nature. So there's ongoing research to try and understand the biology of this a bit better. It seems unfortunate for humans that we are susceptible to Campy infection from any sources, although we see a greater overlap with chickens rather than wild birds. So that's probably a result of exposure. So this is work that I did for my PhD, which is also quite a long time ago now, testing the hypothesis that chickens become infected with Campylobacter from the environment, in this case, wild birds. So we were looking at 2 species of wild birds that are on the university farm in really big numbers. So we wanted to make sure that we captured all of the diversity of Campylobacter in these 2 species. And then we also looked at free-range chickens on the same farm because they're the worst case scenario. They're definitely interacting with their environment. And you can see from the bar chart there that they have very distinct populations of Campylobacter. So then we carried on a bit further with collaborators looking at other wild bird populations. So although we found the 3 species of bird on the same farm had very distinct Campylobacter populations, we found, for example, starlings here from the UK, Sweden and New Zealand have almost identical populations, despite being up to 12,000 miles apart. So it looks as though they might be influenced by the types of feed, the feeding gills that these birds have. And you'll also notice on the left-hand side there that Campylobacter populations from farmed livestock are more closely related to each other, and also more closely related to those that we see in humans. So again, that's probably to do with an overlap and exposure and so on. So another really important thing I think that's come out of the work from genotyping and so on is to be able to use attribution models to sort of model more with more confidence and assign where we think that human disease is most likely coming from, and then be able to do something about it. So this is a publication, a review recently published by Ali Cody, comparing 4 different attribution models, and they fairly consistently say that the greatest contaminated poultry meat is responsible for the majority of human disease in high income countries, although you can see there's some variation there, depending on the methods exactly used and the isolate collections and so on. So I've talked about clonal complexes in C. jejuni, of which is around 40. C. coli seems to be a bit different. So we've known for a long time there's a really big ST828 clade or complex, and that would be the one shown in blue on the left here. And towards the bottom, these are C. jejuni at the top. So at the bottom, the blue clade would be the one that's associated with livestock and farming. There's a couple of other clades shown in red and yellow, and they're a bit rarer, and they've generally been found in environmental waters and sources like aquatic birds, ducks, and so on. It's interesting in the middle there, this panel. So with ST21 as an example, with a C. jejuni strain, where we would see all of the 7 loci derived from C. jejuni, as you would hope, shown in black. And then the one below, ST825, is an example of a C. coli, where all of the alleles are C. coli-like. But we do have examples where we see hybrids. So ST61, known as a C. coli, and that's actually really, really common. That has 6 loci derived from C. jejuni. Sorry, 61, if I said it wrong, is a C. jejuni, but it has 1 UNC allele 17 derived from C. coli. And that's a really common strain type that we see. And then at the bottom there, ST868 is a C. coli. Most of the nucleotide sequence is, we know, is from C. coli, but we do see evidence of genetic import from C. jejuni. So on the right here, this is a model proposed in Sam Shepherd's science paper, where we think that there's 2 species of C. jejuni and C. coli are despeciating. So although they started off at T-2 as the same species and then they diverge, it looks as though, particularly with the clade 1 828 complex in blue there, that's showing increased levels of recombination with C. jejuni in grey. And that's probably driven by intensive agriculture again, and the opportunity for these 2 species to recombine. So for example, you can quite commonly see both species in chicken flocks and also in cattle. And then for the rest of the talk, a lot of the work that we've done is based on a long-running Campylobacter collection, again, going back 20 odd years. We've got the majority of isolates would be from Oxford, but we've also over time sampled in other sentinel surveillance centres. So just to sort of extrapolate what's going on in Oxford and see if that's what's occurring nationally, and the general trend is that it is. So even when we were just restricted to using the 7 locus MLST types with a combination of 3 antigen loci, we found that some strains were persistent over many months. So from this picture, we're not seeing massive genetic instability over time. And since then, this has been extended using the Oxford Core Genome MLST scheme. And here we're able to cluster strain or lineages that vary at less than 5 of the 1343 loci. So we're seeing these lineages are stable. So far, we've seen them stable over up to 17 years. And so most of our isolates are from humans, but we also see the same lineages in other sources such as chickens and ruminants. And then finally, yeah, like everybody else, we've seen increased resistance to particularly ciprofloxacin over time. And then on the right-hand side, this is work that's just been published in the FSA report, looking at enhanced molecular-based surveillance and source attribution of Campy infections in the UK. So we're seeing some differences in antibiotic resistance levels between C. jejuni and C. coli, and then also some differences in the animal sources, which most likely reflects the different use of antibiotics. So to summarise, yes, we have made some progress in 20 years, but of course there's still way more to do. But I think importantly, the MLST, it provides us with a usable nomenclature. So we're able to sort of discuss today what an ST21 is and have some idea of what we're talking about. Attribution studies have been really important so that we know most human disease is derived from contaminated chicken meat in high-income countries, although I haven't had time to go into it, but of course we can't afford to ignore other sources of infection simply due to the sheer scale of human disease infection out there. And then it's been important to investigate the evolution and relationships of Campylobacter between the wild and domesticated animals. It's a zoonotic organism, so obviously it's complicated, don't we know it? And also the long-term monitoring of human disease isolates with the whole genome sequencing has been really, really interesting, looking at trends and sort of increase in or levels of antimicrobial resistance. So of course, this is work that's been done by so many people, but I particularly wanted to mention Ali Cody, who has worked in our group for a long time, but very sadly passed away last year. So a lot of the work that I've done presented today is on behalf of Ali. So thank you, that's the end. Nicole, did you want me to go into the lightning talk?
Thank you. I think Andrew might have some questions for you.
Oh, okay.
Sorry, yeah, thanks, Nicole. Thanks, Fran, for a great history of MLST and bringing us up to date. There have been a couple of questions, and I'm going to start with one from Nabil. There's a bit of chat about this in Discord. Nabil asked, what defines a clonal complex? It's just gone off my screen. Is it quite strict like Salmonella E burst groups? Perhaps you could just give a sort of sentence or two to try and help people along.
So when we set this up a long time ago, we used burst to define the central genotypes. And then when we assign the clonal complexes, we assign the sequence type to a clonal complex when it shares 4 or more alleles with the central genotype. That's the simplest definition. So yes, quite strict.
That's great. One from Anne-Catherine, who thanks for the talk and asks whether the attribution data was based on ST and whether poultry means poultry meat or poultry reservoir.
Very good question. So it's a systematic review of the different attribution models. So there is some variation in there. A lot of it is on the 7 locus MLST types, but there's also some core genome MLST. I'd have to read the paper again. It's usually some variation in whether it's actually chicken meat. I mean, the simplest thing is to say chicken source, poultry source. I think
thank you.
It's largely chickens.
Okay, I'm going to follow. Oh, Arnu's asked, I'm not sure if this is direct to your talk or following on from quite a long chat on Discord, but Arnu's asked, can we combine MLST and AMR studies by specific STs for quinolone and macrolide resistance?
So Desi, who presented earlier, is looking at lineages and how they correlate with the AMR. But we are seeing that there's some correlation, say, with the chicken-associated clonal complexes and levels of ciprofloxacin resistance. Yeah, so I think it's a useful thing to do.
And I'm going to ask one final very quick question. The data for 2021, the frequency for the Oxford data is very low. Is that real, or is that just the study hasn't caught up on itself yet?
Yeah, so that's work in progress. So actually it was a lot higher this year for some reason. There's something weird going on. So that's just because it's partway through the year and we haven't finished it. So yeah, thank you for pointing that out.
Okay, right. Thank you, Fran. I think we're going to have another talk from you now.
Yeah.
So hand back to Nicole.